Hello, colleagues. On behalf of the others, I'm honored to introduce our review, The Plant Microbiome from Ecological Foundations to Precision Microbial Engineering for Sustainable Agriculture. The plant halobiont integrates host, microbiomes, environmental resources, and human governance. Hosting quality genes mediated by directional microbiome-plant interactions on the resources and human regulation, supporting One health, climate resilience, and food security. Guided by this halobiont view, this review outlines a 5-tier pyramid framework from ecology to translation, niche colonization, interaction mechanism, drivers, AM methods, and synthetic consortia, aiming for climate-resilient sustainable crops. At the pyramid base, ecological foundations cover 4 compartments: rhizosphere, phyllosphere, seed, and endosphere. This roadmap guides targeted manipulation. They will examine symbionts, starting with rhizobia. Rhizobia, facultative ligand endosymbionts, cycle through chemotaxis, biofilm, infection, bacterial differentiation, energy-intensive nitrogen fixation, then release upon synthesis to rhizome hypertrophy. We next turn to fungal partners. Complementing rhizobia, a vascular mycorrhiza enhances stress tolerance under phosphorus limitation. Refer4 activates phosphate transporters. During drought, the Zhou-1-Hog-1 mark cascade upregulates aquaporin and Tran halo synthesis. It also regulates ion balance under salt stress. Cross-kingdom communication occurs via effectors and small RNAs. AM fungal SP7 and ECM effectors suppress defense or GA signaling. Fungal sRNAs enter plant cells to cleave immune mRNAs. This multi-level suppression precisely turns immunity to establish mycorrhizal symbiosis. The plant AM fungi's bacterium continue links atmospheric carbon fixation to soil mineral cycles via perivascular space and high phosphere. AM fungi bridge soil and root, creating an integrated ecosystem. This shared pathway enables a comparative analysis between AM and root nodule symbiosis. Both avascular mycorrhizal and root nodule symbiosis employ a conserved signaling model, encompassing Lanxiang, RLK-mediated mic and node factor perception, CSSP-dependent signal transduction, and transcriptional reprogramming. This pathway offers promising targets for engineering nitrogen fixation in non-leguminous crops. The area root mucilage sphere enriches diautrophy and stabilizes communities through carbon allocation, metabolic signaling, and microbial gatekeeping, reframing above ground organs as active nutrient regulators. Then return to phyllosphere. Above ground organs host distinct microbiomes supporting plant fitness benefits. Homeostasis relies on diversity and immunity. Environmental shifts cause dysbiosis and disease. Some internal microbes are seed-transmitted vertically. Endophytes, we see the microbiomes as primary inoculum, assembled via vertical inheritance, horizontal acquisition, and remigration to seeds, forming a transgenerational cycle. Understanding healthy communities is key, as disruptions lead to disease. This leads us to pathobiome concept. The one disease, one pathogen paradigm is revised. Few diseases often result from pathobiomes. For example, tomato wilt with 19 Fusarium species, and soybean root with cross-kingdom partners. Future management should integrate host and environment into whole network microbiome strategies. Microbiome assembly is driven by host filters and environmental filters. Host sets filtering direction. Environment defines the microbial pore and boundaries, shaping a continuum from box soil to endosphere. This foundation enables precision manipulation. We propose an integrated pipeline, absolute abundance quantification, multi-omics association linking genotype, microbiome, and phenome, and precision syncom application to close the loop and design synthetic communities. Syncoms are modular, controllable, and robust, built via top-down and bottom-up approaches. Key challenges: biosafety, native microbiota resistance, and evolutionary stability must be addressed to bridge lab design to field application. High-throughput sequencing and multi-omics are core, from data to key taxon and functions. Emerging technologies such as DNA-seq, single-cell RNA-seq, and spatial transcription omics. This brings us from lab to field, translating research into standardized products. Currently, inoculants are moving from single strands to simplified syncoms. Field stability can be enhanced by pairing prebiotic cohesion compounds with crop recruitment genes. This concludes our journey from ecology to application for sustainable agriculture. Thank you for your attention.